Welcome, everybody, to the workshop today. My name is Steve Williams from Thermo Fisher Scientific. Title of today's workshop is Testing for HIV Integrase Inhibitor Resistance 2024 and Beyond. So this workshop is specifically designed for public health and clinical laboratorians, as well as healthcare providers who wish to learn more about the current and the future state of HIV resistance in the area of integrase strand transfer inhibitors, or INSTIs as they're more commonly known. In this workshop, we'll address the importance of understanding HIV drug resistance at the population and at the public health level, and at the individual or the clinical level, and to also understand the impact of HIV drug resistance on the effectiveness of antiretroviral therapy for HIV treatment and prevention. So HIV drug resistance can emerge with the use of antiretroviral drugs for HIV treatment and prevention. And HIV drug resistance has the potential to reduce the efficacy of antiretroviral drugs, leading to an increase in the HIV incidence, morbidity, and mortality. The increasing prevalence of drug-resistant HIV strains poses a serious threat to the progress made over the past few decades in combating the HIV pandemic, and can be attributed to various factors, including incomplete adherence to antiretroviral drug regimens, suboptimal drug availability, and the transmission of drug-resistant HIV strains. So the World Health Organization estimates that approximately 10% of people initiating ARV or antiretroviral treatment globally have drug-resistant HIV strains. And to combat this issue, the WHO recommends HIV drug resistance monitoring and surveillance as an integral part of comprehensive antiretroviral therapy and pre-exposure prophylaxis PrEP programs. So the presentations from our speakers today will bring together a specific focus on INSTIs for HIV prevention and treatment, and will bring forward the perspectives of our speakers who are leaders in the field of HIV drug resistance, and they will discuss the current state of drug resistance globally, the use of HIV drug resistance testing for public health and surveillance, and clinical management of persons living with HIV, as well as new challenges related to detection and monitoring of HIV drug resistance. We'll also explore the challenges and considerations surrounding HIV drug resistance among populations receiving PrEP to prevent HIV acquisition. So hopefully, by the end of this presentation, you will have a comprehensive understanding of the current state and challenges presented by INSTI resistance and its implications for public health and clinical laboratorians for those directly involved in HIV treatment and prevention. So let us begin our journey into this important topic. I'm honored to introduce the first speaker, Dr. Gert van Zyl. He is a virological pathologist and professor in medical virology at Stellenbosch University, and the executive head of the Department of Pathology, also at Stellenbosch University, and National Health Laboratory Service, Tygerberg in South Africa. Dr. van Zyl studied medicine at the University of Pretoria and specialized in virological pathology at Stellenbosch University. He obtained his PhD investigating HIV drug resistance in 2012. And this involved the investigation of drug resistance in pregnant women, adults and children exposed to successive treatment regimens used in South Africa public sector. His research focus areas are HIV drug resistance and HIV persistence. Other research areas are third-generation sequencing and viral diagnostic test methods. Much of his research relates to diagnostic service delivery in resource-limited settings, which often require novel solutions. So welcome, Dr. van Zyl.
Thank you very much, Dr. Williams. It's a privilege to be here. So the disclaimer, what I want to cover in this talk are the following: antiretroviral drugs and genetic barrier, acquired versus transmitted resistance, HIV drug resistance testing, the public health importance of HIV drug resistance, surveillance strategies, and then current gaps in knowledge and future perspectives. One might ask, why is it that we are able to treat HIV so successfully? It is because there are various steps in the viral life cycle or replication cycle for which there are inhibitors available that we can use in combination to block viral replication and get patients' viral loads down. The first step in replication that one can target is entry, but there are few entry inhibitors used in current therapy. The next important step is reverse transcription, where one has NRTIs, nucleoside reverse transcriptase inhibitors, and non-nucleoside reverse transcriptase inhibitors. After reverse transcription, double-stranded DNA is integrated into the host cell's DNA, and this is catalyzed by the enzyme integrase. And integrase strand transfer inhibitors inhibit this step. Then, once viral particles have formed, they're not infectious. They have to undergo further maturation. And that is catalyzed by protease, again inhibited by protease inhibitors. I want to highlight just a few of the important drugs that you will encounter a lot. Of the NRTIs, tenofovir is available as two different prodrugs, disoproxil fumarate, or TDF, and then alafenamide, or TAF. And it's often combined with either 3-TC, lamivudine, or emtricitabine, FTC. Of the NNRTIs, I want to highlight efavirenz, which has been the mainstay of treatment, of first-line treatment, until 2019. Of the protease inhibitors, I want to highlight darunavir, which we use a lot in treatment-experienced patients. If one looks at the INSTIs, then there has been a transition to second-generation INSTIs, where dolutegravir is used in many countries in the world now as the mainstay of first-line therapy, and bictegravir and cabotegravir are also used. Cabotegravir is available as a long-acting drug, which is injectable, used for prevention and treatment. So what is acquired drug resistance and what is transmitted resistance? Acquired resistance emerges in a patient on therapy due to the evolution of the drug in the presence of drug pressure, whereas transmitted resistance is when somebody is infected with a resistant variant transmitted from another patient. Detecting transmitted resistance is often impractical because one has to test very soon after infection. The alternative, which is more pragmatic, is to test patients when they start therapy, and this is called pre-treatment drug resistance. So why is it difficult to detect transmitted resistance? Even if a patient is infected with one variant, which is often the case, and that variant has resistance, once this variant starts to evolve and revert to wild type, these wild type viruses often replicate a lot better than the original resistant variant and could displace it in the population. So if you test too late, you may not be able to detect transmitted resistance. Variants with low fitness are also unlikely to be transmitted from one generation to another. However, there are certain mutations would do not result in a huge fitness price, and these variants can be transmitted from one patient to another through successive generations and thereby spread resistance rapidly. A very important concept for us when we consider drugs is the concept of genetic barrier. That basically tells us how difficult it is for drug resistance to evolve. A drug like darunavir requires multiple mutations before you have a high level of resistance. And these mutations often also come at a fitness price to the virus. Low genetic barrier drugs like efavirenz or nevirapine, in their case, one mutation causes a high level of resistance, and you can no longer use these drugs to treat patients. What is an HIV drug resistance test? People may be more familiar with the drug resistance testing that's done for bacteria, where one would plate bacteria on an agarose plate and look for a zone of inhibition around an antibiotic disc. We cannot do that for HIV because the patient's virus might not even grow in your cell culture system. For that reason, we first amplify the gene of interest, and then that is recombined with a backbone that is culture adapted. This chimeric construct is then grown in the cell culture and exposed to different concentrations of different drugs to assess susceptibility. You can imagine that this process is costly, time-consuming, and therefore not widely used. Once we know which mutations are associated with drug resistance, we tend to prefer to do genotypic resistance testing just for practical reasons. So how is that done? This will be described in a lot more detail by Dr. Parikh in her talk. But to simplify this, a sample is taken from the patient, and from the plasma, we isolate RNA. This RNA is then reverse transcribed to DNA, which is amplified by PCR, and then sequenced through various means. And this nucleotide sequence is then interpreted through a drug resistance database. And what you get when you want to care for your patient is you get a report, and this report tells you which drugs your patient's virus is resistant towards. And you can then use that to choose appropriate therapy. A very important concept is the so-called U equals U. Undetectable means untransmittable. Using antiretroviral therapy to suppress population viral load is the best proven biological intervention to prevent HIV transmission. And therefore, the UNAs in their 2030 95, 95, 95 targets emphasizes this. They want 95% of people knowing their HIV status. Of these, at least 95% should be on treatment, and of these, at least 95% should have suppressed viral loads. What is the impact of drug resistance on these targets? It especially threatens the last of the 95s. Because if people have transmitted drug resistance, it would impact on the success of your first-line regimen. These patients are also then likely to require a second-line regimen. They have to be switched. And again, there's another opportunity for resistance to evolve. And depending on the fitness of these variants, they can be transmitted to others. So drug resistance is important for us in terms of public health and patient-level management. And these two things are often integrated. It helps us to answer the following questions. Is a regimen likely to succeed in a patient based on what you expect in the population? And it also tells us how to monitor patients. If you don't expect a high probability of resistance, then in resource-limited settings, a resistance test may not be rational or warranted. But what we've seen with efavirenz therapy is that when patients fail this regimen, the risk of resistance is so high that in public health settings and resource-limited settings, patients warranted a switch to second line without needing a resistance test. There has been a recent need to change guidelines, and this is due to three main reasons. The first was there was an increase in pre-treatment NNRTI resistance. The second was that dolutegravir-based therapy has been shown in multiple trials to be either superior or non-inferior to efavirenz-based regimens. And lastly, when patients fail a dolutegravir regimen with high viral loads, and that's used as first-line therapy, there's a very low probability of resistance. And patients can be resuppressed on these regimens without needing to switch them. So because of that, in 2019, the WHO introduced dolutegravir-based therapy as their recommended first-line therapy when combined with 2 NRTIs. And in 2021, they've also introduced it for second-line therapy. So what are the WHO guidelines? Dolutegravir in combination with 2 NRTIs is the preferred first-line ART and the preferred second-line ART after NRTI regimen failure. In practice, in resource-limited settings, generic fixed-dose combinations are used because of ease of use and low cost. Tenofovir combined with lamivudine and dolutegravir is referred to as TLD, and when it's used for first-line therapy, we call it TLD1. When it's used in treatment-experienced patients, we call it TLD2. This resulted in second-generation INSTIs, especially dolutegravir-including therapies, predominating worldwide in therapy. Not only the treatment landscape has changed, but also the pre-exposure prophylaxis landscape. Injectable cabotegravir has been shown to be superior to oral pre-exposure prophylaxis. Breakthrough infections are rare, but when they do occur, there's a high risk of resistance. We don't know what the risk factors for these breakthroughs are, but it's important for us to diagnose these cases early and to exclude patients who might be an acute infection from starting PrEP. And to do that would require viral load testing to detect early acute infections. One challenge with CAB-LA is when patients are stopped on these drugs because of their very long half-life. For months after that, there could be significant levels of these drugs present in their blood and tissues. And should they then become infected, there's a high risk of resistance. So we need an approach to manage these patients. There's also more data needed on transgender individuals and what the dosage should be in these patients. The WHO recommends several ways to do surveillance for drug resistance. They recommend nationally representative laboratory-based studies, clinic-based studies, and sentinel laboratory studies where the same lab is used sequentially to look at trends. And they also recommend surveillance in PrEP breakthrough cases. Priority groups are newly diagnosed adults, newly diagnosed infants, and in treatment experience and adults and children with virological failure, and also PrEP users. The International AIDS Society has identified several mutations that are associated with dolutegravir resistance. It's listed here. The ones bolded are the major mutations. And there's also evidence from a study by Bob Schaefer's group that showed that when patients start with dolutegravir as their first INSTI, in particular those 3 circled mutations are the most common. The treatment success on dolutegravir regimens and the risk for resistance are inversely correlated. And in the different scenarios, there's a higher or lower risk. When patients are treatment naive, there's a very high success rate, and we really don't see drug resistance in these patients. When patients transition from a first-line regimen to dolutegravir, and this is done often in resource-limited settings without doing viral load testing, when they are viremic, there's a higher risk of resistance and also a higher risk of failure. Treatment-experienced patients with proven virologic failure would transition from an efavirenz regimen to second-line, still have a high treatment success rate, but now that the risk of drug resistance when they have virologic failure becomes high, or relatively high. Integrase experienced patients, when they transition to dolutegravir, there's a high risk of failure and of drug resistance. There are different ways to study acquired HIV drug resistance. Randomized controlled trials are beneficial because allocation is unbiased. But what are the problems with randomized trials? They have strict inclusion criteria, so patients with advanced disease would be excluded, And because it's costly, follow-up is also often limited. So what do we then need? We need population-based studies to look at longer-term outcomes. And that would also provide us with information about patients with advanced disease. Laboratory-based studies also have their advantages and disadvantages. What are their advantages? They're low cost, and one can use them to study longitudinal trends, but they have referral biases. But if one uses the same approach year after year, it's valuable because one could then see if there's an increase in your patient population. So to summarize, what do we know about dolutegravir-acquired resistance? Prevalence is very low when patients start on a combination of dolutegravir-2 NRTIs and are treatment naive. When they are treatment experienced, such as patients previously using efavirenz and switching to this regimen, less than 2% will develop resistance. But when they have virologic failure, about 20% will have drug resistance. With reference to pre-therapy resistance, what do we know? Large studies have been done, and there are also systematic reviews. And there's no evidence that there's an increase in transmitted integrase resistance and also affecting dolutegravir. So what are the future directions? What do we need to know? We still don't have a good grip on what pattern of adherence is associated with dolutegravir resistance. We also don't know what are the high-risk subgroups. We don't know what the population impact of CAP-LA would be for prevention. And longer term, we need to keep on looking because we don't know what the trends of transmitted resistance would be in future. And the question can be asked, how durable is it to use integrase transfer inhibitors in first-line therapy, second-line therapy, and for PrEP, for prevention? So I want to conclude with the following question. Is INSTI first-line therapy currently threatened? There's no evidence that it is, but there are multiple unknowns. For many of the mutations that we see emerging, we don't know how transmissible they are. We don't know how future changes in drug regimen use would impact this. And we don't know what will happen with a widespread use of CAP-LA for prevention. The important thing is to realize that INSTIs are programmatically very valuable. We cannot afford to lose them. So although there's a short-term low risk of losing them, we should be prudent and should continue with surveillance. Thank you.
Thank you, Dr. van Zyl, for that interesting view on HIV drug resistance from a public health perspective. Would like to move on to our second speaker. So I am pleased to welcome Dr. Rami Kantor. He will provide a clinician's perspective on HIV drug resistance. So Dr. Kantor is a physician scientist at the Division of Infectious Diseases in the Department of Medicine at the Brown University Alpert Medical School. And as a clinician, he cares for patients living with HIV. And as a researcher, he directs the Drug Resistance Laboratory at the Providence-Boston Center for AIDS Research and co-directs the CFAR Basic Science Core. He is the past chair of an NIH AIDS study section and a member of the Department of Health and Human Services Guidelines panel for HIV treatment in adults and adolescents, and deputy editor at the Journal of International AIDS Society. His clinical and basic science research focuses on HIV drug resistance, treatment monitoring, and transmission through networks in diverse global populations and settings. He has multiple collaborations with clinicians, molecular, computational, and evolutionary biologists, statisticians, engineers, and behavioral scientists at Brown University, both nationally and internationally. His interests are multidisciplinary and include basic science, translational, clinical, and bioinformatics research, and he is the author of over more than 300 publications. So welcome, Dr. Kantor.
Thank you so much for this kind introduction. So I'm going to talk about the clinical perspective of HIV integrase inhibitor resistance. This is my disclaimer. And the objective of my talk will be to learn about the use of HIV drug resistance testing for clinical management of people living with HIV in the era of integrase inhibitors. I'm going to talk about a little bit of introduction and on clinical drug resistance, important for us to understand how to treat patients. We'll talk about the current landscape of drug resistance and its impact, and how they are incorporated into the guidelines and the clinical implications of that landscape and how it's integrated into routine clinical care. We'll describe a case study to see how this is implemented and talk about gaps and future directions, and conclude with some key takeaways. So here's a disease treatment resistance timeline for someone who is living with HIV. So at some point, someone is infected. We usually don't really know when that is. Then at some point after that, they're diagnosed. And at some point after that, they'll start what we call first-line therapy. And then if they take it throughout their life and they're adherent and everything is great, that might be the only treatment they take. But sometimes things happen and they'll fail their first-line therapy and will have to go to second-line therapy, and so on and so forth. They might fail their second line, or they might continue it, and then they might need to go to third line, and so on and so forth. So before we start treatment, and we heard in the prior talk, someone might already have resistance that they got from the person that they got the HIV from. We call that transmitted resistance. And the resistance that develops while they are on therapy is called acquired resistance, and we can talk about first-line resistance, second-line resistance, and so on and so forth. The general concept, without getting into too many numbers, is that few mutations are transmitted, few mutations are developed at first-line therapy failure, although that asterisk says that if you fail for long enough and you allow mutations to develop, they will develop, so it can be accumulating. And then in more advanced treatments, more mutations will develop, and that might impact the regimens that we can give our patients. If we talk about mutations for a second, this is a schematic showing an enzyme with an active site in the middle, and that's where usually the drug target is and where the drug will work. And the mutations that develop are related to this. So there's a class of mutations that we'll call major mutations, and you saw a list of some of them. And those are mutations that are usually close to the active site or around the active site, and their impact is the highest. And then there are mutations that are more minor, more farther away from the active site, and they might either compensate for the major mutations or add some additional resistance. And this concept is true for all the drug targets that we have today. I'm showing them on the left, including integrase, and I'm showing you some of the integrase mutations that you saw on the prior list and how they are located in terms of the closeness to the drug target active site. Another important concept is the barrier to resistance that we have to think about when we're treating patients. So on the x-axis, you could see the number of mutations. On the y-axis is their implication on viral replication. And basically, there's a group of drugs that will have a low barrier to resistance, meaning that you need a low number of mutations in order to have a resistance impact with a very low implication on their viral replication. And there'll be a group of drugs that still require a low number of mutations but might have a better impact on the viral replication. And then you'll have the best drugs from our perspective, which is you require a lot of mutations to create a high impact resistance, and they are very impactful. So if we try to think about the drug classes that we have today, and this again is part of the thinking when we're designing regimens, the NRTIs, the oldest drug that we have, are usually thought to be on the lower left of this graph. The NNRTIs and the first generation integrase inhibitors that I'm showing on the left, raltegravir and elvitegravir, are thought to have low barrier to resistance, but these mutations might be a little bit more impactful. And then the best drugs with the highest barrier to resistance are the second generation integrase inhibitors that I'm showing here on the right, dolutegravir and bictegravir. Cabotegravir is with an asterisk because I would say we still are learning about the drug resistance barrier of this drug, and also the protease inhibitors that are thought to have a high barrier to resistance, and it's really hard to develop resistance to them. So another concept important is the regimens that are available to us, and then what I call the dogmas of how we treat patients. And I'm showing the timeline on the left, and here's along the timeline the different dogmas that we had. We started from the early days where we would give either one drug or two drugs, what we'd had available then. We went to the mid-90s where the cocktail came to be, or the highly active antiretroviral therapy, with giving 3 drugs from more than one class to treat HIV. Then we had a single tablet regimen, which is a big deal for people who are taking multiple drugs for the rest of their lives. And we had non-single tablet regimens, but less pills taken for people who have more advanced disease or complex resistance. And then more recently, we're going back in time, and we're treating people with 2 drugs, and now with long-acting therapy drugs, and that's because our drugs are getting much better. Part of them is the integrase inhibitors that we're talking about today. This last couple of years where we have these change in dogmas are a big deal for people who are living with HIV and for people who are treating them. If we talk about the timeline related to integrase inhibitors that I'm showing on the right, this is the first generation integrase inhibitors, and then green are the second generation integrase inhibitors. You can see that they're more recently incorporated into our lives, but they are incorporated throughout these new treatment dogmas. So basically today when we're treating patients, integrase inhibitors are all over, and we have to think about all these concepts that we're discussing here and when we're treating regimens. If we talk about the current landscape of resistance, thinking about these dogmas, whether it's transmitted resistance on the first line or all the rest, which is acquired resistance, and then again, conceptually, transmitted resistance is pretty high. We could see places where it goes up to 25% of people who have acquired new HIV who already have transmitted resistance. The early days showed us that a very large proportion of people who are treated with these 1 or 2 drugs that were older with lower barrier to resistance, like we saw in the other graph, the resistance to them would develop quickly and in very high proportions. But when we start going down with time and we inserting the cocktail, the single tablet regimens, the 2 drugs, the long-acting therapy, all with integrase inhibitors highlighted in yellow here, we can see that the arrows narrow. So the resistance develops pretty rarely. A little bit more when we talk about more advanced people, but in general, these drugs with these high barrier, these regimens with high barrier drugs will develop less resistance than what we were used to earlier. So if we think about the current landscape of drug resistance or the trends, again, this is time on the x-axis and drug resistance on the y-axis. If we look, since we started doing drug resistance testing in the early 90s or late 80s, any acquired resistance is going down from around 70 to 80% to around 40 to 50%. And if we break this acquired resistance down to which medications kind of drive it, we could look at the NRTIs, which is a line that's kind of parallel to the any-acquired resistance that is still there, but going down with maybe a little bit lower shape. The NNRTIs drive this a lot because this used to be a very highly used class and resistance to it developed pretty quickly, but it is coming down because we're using them less. Integrase inhibitors, you see that they started later because this, again, like we discussed, is a more recent class. They're lower, but they're stable throughout the years that we have seen them. And then PIs started higher, medications got better, and they're lower because they're also used less because INSTI are used much more. And then transmitted resistance is actually going higher throughout the years, mostly again driven by NNRTIs like we've been discussing, And what happens in the more recent years, I think, still needs to be determined. What is the impact of all this resistance? Well, the thought is that drug resistance leads to failure. That's why we have treatment failure and viral non-suppression. So we have to think about this, and we have to treat people that have drug resistance so that they will have a viral suppression in a sustainable way. The asterisks means we're still learning about this, and I'll come back to this when we talk more about the impact of integrase inhibitors. So these concepts are inserted into our guidelines. So here's the timeline that we saw before, and what our guidelines tell us today is to do drug resistance testing before someone starts antiretroviral therapy to look for transmitted resistance, and then every time that they fail, whether it's first line or second line or later, so that we can identify the resistant drugs and design a regimen based on that. The guidelines also tell us that integrase inhibitors are throughout someone's treatment trajectory. So they're recommended as first line, as second line, and as more advanced lines of treatment. We already heard that resistance to integrase inhibitors are rare for transmitted resistance. The asterisk says, well, we're not really looking for this. We'll talk about this in a second. We also heard that drug resistance to first-line therapy is rare. I'm putting an asterisk here because, again, this is something that is emerging as maybe not as low as we thought it is, so this is something we definitely have to continue to monitor. And then maybe it's more common in more advanced situations. So as a result of these concepts, our drug resistance testing recommendations in developed countries' guidelines say you don't have to look for transmitted resistance for everybody. Just look for it when you think that it might be high. In developing countries, really we're only looking at drug resistance in the more advanced situations on an individual basis for individual care because this is just not feasible otherwise. So when we try to design a regimen for our patients, we have to take these concepts, the drug resistance barriers, the mutations, and the different dogmas on how to design a regimen. And we have to remember to add the patient perspective or considerations. And I'm showing you a couple here. I won't go through them. It's a long list, but just going sort of clockwise, whether someone takes their treatment adherence is very important. How many pills throughout the day? Did they have past resistance before this current regimen? What was their past treatment? Did they fail in the past and for how long? What is the viral load monitoring capacity and where do they live? What are their comorbidities, mental health issues, housing or life circumstances that might impact their taking their medications, food interactions, drug-drug interactions with the antiretrovirals, and what other drugs in the regimen is there. So there are a lot of considerations that we have to take into account, and they all impact INSTIs. I'm highlighting in red just some of those. You might need to take more than once a day integrase inhibitor if you're at a more advanced stage. Definitely important, your past resistance and past treatment information and your interaction with other medications in the regimen. So regimen design really, we're thinking about medicine and there are debates on whether medicine is art or science. I would say that for drug resistance testing and regimen design, it's definitely higher on the art. And we have to take all these considerations when we design a regimen, and it definitely gets more complex with time because this drug resistance can accumulate. So other tools that are available to us when we think about what regimens to design, we heard a little bit about this. So we could do genotyping, drug resistance testing, phenotyping, and we have to remember about accumulated resistance and archive resistance. I'll go through these very briefly. Genotyping, as we already heard, we sequence the virus, we translate it to amino acids, we derive the relevant mutations, and then we determine what their impact might be to our patients. In red are medications that are high-level resistant, in orange sort of middle, and in green sensitive, and we design a regimen based on that. We can also do phenotyping, which is actually measurement of drug concentrations and of fold capacity of the virus and its ability to overcome this resistance, with a bottom line also of this red orange, green list, which is which medications can we give to our patients. And then we have to remember that with time, this resistance accumulates and it can be archived in the proviral DNA and then reemerge in the right setting. So all these are things we take into account, and our guidelines tell us that we have to let drug resistance testing guide us, and the general concept is to take a high barrier to resistance drug plus one or two other active drugs in the regimen, and we have to use all of these tools to guide us on regimen design. So if we just take an example of how this would be, one high barrier drug plus two active drugs could be a bictegravir from the high barrier second generation integrase inhibitors plus two NRTIs. In this case, I'm showing you TAF and FTC, and this would be probably the most common treatment regimen that we're starting people in developed countries today. Another example could be dolutegravir, again, from this high genetic barrier circle with two other NRTIs, and this is the TLD that we heard about, which is the most common drug that people are starting today throughout the world in developed settings. And we could use the same concepts to design a more complicated regimen with maybe two drugs from this high genetic barrier circle with two NRTIs, and if needed, even another second generation NNRTI, again, depending on the resistance patterns. So let's think about a case. So here again is our timeline. This patient, we don't know when they were infected, but they were diagnosed in 2004. They started first-line therapy in 2006, and they received a whole bunch of medications throughout the timeline as we've been discussing. It's unclear if they were exposed to raltegravir, which you remember is a first-generation integrase inhibitor. And throughout these years, they had high viral failure. They had a viral load of greater than 100,000 more than 5 times. And then at some point in 2017, they were able to get a resistance test. And this resistance test showed 4-class resistance. 1, 2, 3, 4. I'm showing you the specific mutations. This is high-level drug resistance. And here is the impact of that drug resistance. Again, red drugs show basically medications that you're not supposed to give because you have resistance to it. Orange is sort of intermediate, and then green is susceptible. So thinking about the guidelines, which is high barrier drug plus 1 or 2 active drugs, it's really hard to design a regimen, but what this patient received eventually is 2 medications with high genetic barrier to resistance that both have intermediate resistance, as you'll see. They received tenofovir, which is susceptible, which is an NRTI, and they received 3TC, which is high-level resistance, but has a specific mutation that has a fitness impact. And with this treatment, the patient became undetectable. So really the question is, well, what does dolutegravir resistance mean if we know that it has intermediate resistance? But when we design a regimen that might be good enough, the patient can become undetectable. So let's talk a little bit about existing gaps in the field. I think one which I kind of alluded to is, the newer the drug on the x-axis, the less we know about it. And that impacts our understanding about HIV drug resistance and integrase inhibitors that are more recently introduced. And the treatment regimens are heterogeneous. So here's a line of drugs that people can receive throughout time, but how they receive it could be in a different way. They could receive only one regimen throughout. They can be, historical with many regimens throughout, et cetera, and so on and so forth. And integrase inhibitors are all throughout this, these treatment regimens. And if we add the cabotegravir that we heard, excuse me, will also be entering in terms of prevention, we could see the complexity of this with time as we think about people's drug resistance patterns. Another existing gap is what I call the global burden paradox. So in developed settings, non-low middle income countries, we generally have available genotyping, phenotyping. We can look at accumulated resistance with past resistance. We can look at archive resistance, and ART is almost unlimited. Maybe insurance issues are still problematic, but in general. This is not the case in low and middle income countries, where we have genotyping sometimes, and only in advanced patients, and maybe only in a population health, public health approach, but we really don't have all these other ways to assess resistance, and we're limited by the medications that we're giving. This is a major gap. And then there are other gaps. I won't go through them with time. Different populations that are important, the fact that integrase are throughout the lines of treatment, how they interact with new medications classes, some discordances that we need to understand better in terms of resistance. We didn't even talk about cost and cost effectiveness. And then there's also HIV-2 globally and other parameters that we'll hear more about in the next talk that we need to consider and to address as we think about this. So we have to address these gaps. We need to think if we need new approaches to our thinking and regimen design as these new classes and advanced medications, which are incredible, are incorporated. And maybe we need to be more forgiving for resistance, like the treatment case that I showed you before. We just don't know that. So this is where this comes back, which is, well, what does really resistance mean? Does it really need to lead to failure? What type of resistance needs to failure, and how do we think about this? This is an important gap. So to conclude, we have excellent antiretroviral therapy anchored by integrase inhibitors that save lives of people with HIV. Despite extensive knowledge, HIV drug resistance remains a hurdle to long-term therapy. Sustainability and testing for it is essential for individual care. Clinical management of diverse forms of HIV drug resistance in the era of INSTIs is an art integrating multiple dimensions. The global paradox related to HIV drug resistance, testing availability, and other existing gaps should be urgently addressed. Longitudinal data are essential for new and emerging drugs and formulations, including integrase inhibitors, as their use expands in both treatment and prevention. Until further experience is acquired, close monitoring, adherence to guidelines, and seeking expert advice are recommended. And the time to address the challenges of integrase inhibitors and other drug resistance is now, ensuring that it does not undermine the progress made in HIV drug therapy. Here are some references that might be important and some relevant resources for drug resistance, and thank you so much.
Thank you, Dr. Kantor, for walking us through some of the important considerations in treating people infected with HIV. Our final speaker for today will be Dr. Urvi Parikh. She will focus on the laboratory perspectives of HIV drug resistance testing. Dr. Parikh is an associate professor of medicine at the University of Pittsburgh, Department of Medicine, Division of Infectious Diseases. Her research focuses on examining the risk of HIV-1 drug resistance with the use of antiretrovirals for HIV prevention. She co-leads the ACTION and SeroPrep studies investigating causes of HIV acquisition in individuals using PrEP, and she is the Associate Director of the AIDS Clinical Trial Group, ACTG, at the University of Pittsburgh Virology Specialty Lab, and is the HIV drug resistance technical lead of the USAID PEPFAR Maximizing Options to Advanced Informed Choice for HIV Prevention, or MOSAIC, project. She was also the Associate Director of the Microbicide Trials Network, MTN, Virology Core, and has served as a protocol virologist for studies evaluating the safety and efficacy of candidate antiretroviral products for HIV prevention. The laboratory conducts sensitive assays to diagnose genotype and phenotype HIV-1 and conducts preclinical research towards the development of new agents for HIV prevention. Welcome, Dr. Parikh.
Thank you. I'll be speaking to you today about current and evolving methods for HIV drug resistance testing for integrase strand transfer inhibitors. So here's my, is it advancing? Okay. Here's my disclaimer. The learning objective for today will be to get updated on current and evolving methods for HIV drug resistance testing and the laboratory challenges posed by the Integrase strand transfer inhibitor class of HIV inhibitors. So I'll cover several topics today. One is to give you an overview of the current landscape of HIV drug resistance testing methods and their specific uses. Second, I will explain the principles and methodologies of current and emerging HIV drug resistance technologies, and we'll give you some examples of some clinical or research applications that use these technologies. I'll talk about some of the advantages and limitations of the different HIV drug resistance testing approaches, and then conclude with some key takeaways on the current and future state of HIV drug resistance testing methods. There are three major indications for HIV drug resistance testing that are summarized here. Clinical indications, as recommended by the United States Health and Human Services, include testing for entry into care to guide regimen selection with consideration that ART should not be delayed awaiting test results, especially in pregnant people with HIV. Individuals with virologic failure or suboptimal viral load reduction on their current regimen should also receive an HIV drug resistance test. Surveillance is another important indication for drug resistance testing. Globally, surveillance may be done on dried blood spot specimens, and results may not always go back to the individuals, as testing turnaround time may be insufficient for the result to be clinically relevant. Two types of HIV drug resistance surveillance recommended by the World Health Organization include surveillance for acquired drug resistance or resistance that develops after treatment, and pre-treatment drug resistance, which is resistance that includes transmitted resistance from a partner. Surveillance has been routinely done for reverse transcriptase inhibitors, NRTIs, and NNRTIs, and is currently expanding for INSTIs. For each of these indications, there are commonly used assays, which will be described in more detail in this talk. For clinical management and population surveillance, typically bulk or Sanger sequencing is the most commonly used assay type. Next-generation sequencing is emerging for clinical management, but is still largely used for research, with research applications including whole genome sequencing, single genome sequencing, and newer technologies like long-read sequencing and point mutation assays. This slide illustrates the overall principle of all HIV gene sequencing. Essentially, HIV RNA is extracted from plasma using chemical or bead-based methods, followed by reverse transcriptase to DNA, then amplification by PCR. The amplification step can be tailored to gene regions of interest. Typically, a minimum of 200 copies of RNA per milliliter of plasma is needed to have sufficient template for amplification, but some research applications can go lower. The amplification products are purified and sequenced using various methods. So I'll start with the oldest technology, which is the bulk or Sanger sequencing. It's still the most common method used for genotyping clinically. This type of sequencing uses primers that cover specific regions in HIV, focusing on the three regions that correspond to the drug classes most frequently used for antiretroviral therapy, including protease, reverse transcriptase, and integrase. Sanger sequencing follows a typical workflow as described earlier. So here, individual forward and reverse sequencing primers are needed to cover the gene regions of interest, with protease RT usually done as one assay and integrase done as separate assays, as shown by the gels from our lab here. The end result is an electropherogram, which depicts each base as a separate color, which can be manually read or analyzed using software. The process can be labor-intensive due to the many manual steps, and the mutation threshold detection is typically 20% frequency. However, there are well-established external quality assurance programs and clinically approved kits making this method still widely used for clinical sequencing. So illustrated here is a recent application of Sanger sequencing, which we presented at a conference earlier this year. Sanger sequencing is an established method in many settings because of equipment infrastructure that's already in existence, and this can allow secondary analysis using data from routine care. So in this case, remnant samples from Western Kenya were sequenced to compare the frequency of INSTI resistance in individuals who started dolutegravir-based first-line antiretroviral therapy or switched to dolutegravir to use a second or third-line antiretroviral therapy. The main findings of this study are shown in the last row. So of 12 individuals who are on first-line dolutegravir-based ART with a successful sequence, only 1 of 12 individuals, or 8%, had INSTI resistance. By contrast, of 31 individuals on second or third-line dolutegravir-based ART, 7 or 23% had INSTI resistance. This difference was significant and indicated that the risk of developing dolutegravir drug resistance mutations may be higher in those with a background of pre-existing mutations. And in this case, using Sanger was an effective tool to do this kind of analysis. One of the challenges of Sanger is that individual bidirectional sequencing primers must be used, one forward and reverse for each gene region, and are individually read and analyzed. So for example, 6 individual sequencing reactions are needed to cover the integrase gene. This can make the process and analysis labor-intensive. So newer technology of next-generation sequencing, or NGS, can increase throughput and sensitivity through massively paralleled reading that are simultaneously sequenced. This allows barcoding and pooling of samples to generate data more efficiently. The workflow of NGS, or next-generation sequencing, is depicted here in this figure on the left. So the first step is that the PCR-amplified sample is processed into short fragments and ligated to adapters to form a library. The adapters have a unique sequence that serves as a molecular index, or barcode, which allows multiple samples to be mixed and sequenced at the same time. Then the library is attached to a solid surface like a flow cell, and each template is PCR amplified on the flow cell to ensure optimal signal for the instrument. Then third, sequencing by synthesis allows all samples to be read simultaneously as the instrument used detects the newly incorporated bases. And finally, the collected data processes fluorescent signal into base calls, which is assembled into sequencing reads and assessed for quality. Samples are demultiplexed according to their index or barcode, and each set of reads are aligned to a reference sequence to identify resistance mutations. So this type of technology is higher throughput. There's a potential for lower costs for high sample volume laboratories, and there's a higher sensitivity for calling mixed bases. So in Sanger, the threshold was about 20%, but here you can accurately go down to 5%. But you do need bioinformatics for data analysis. This cannot be analyzed manually. An additional research application for NGS is the use of unique molecular identifiers, or UMIs, to increase sensitivity and accuracy of mutation detection. So this is kind of a cool technology where you use these UMIs to allow bioinformatic correction of PCR and sequencing error. So here's an illustration of how UMIs work. Say you have a donor sample from an individual that has 3 mutations on different genomes, each at 33% frequency. In routine NGS, due to inherent biases in PCR, say genomes with mutation 1 amplify most efficiently, while genomes with mutation 2 amplify least efficiently. So this would result in mutation frequency calling of 60% mutation 1, 10% mutation 2, and 30% mutation 3. So if you instead take this 10-mer unique molecular identifier and barcode it at the reverse transcriptase step, it would tag each cDNA molecule and result in individual RNA resulting from each individual RNA template. So following amplification and sequencing, templates with the same UMI can be bioinformatically collapsed to yield accurate quantitation of the number of starting genomes. And this gives you an accurate quantitation of HIV drug resistance mutations. So to be clear, non-UMI or standard NGS application can be very accurate, often down to 5% mutation frequency, but the quantification can be less certain. So this particular application of UMI NGS is important if you need to identify very low frequency mutations down to 1%, or if you want to determine if multiple mutations are on the same genome. So now I'll go into several of the research applications of NGS. So one application would be single genome sequencing. This is where limiting dilution is used to sequence individual genomes, and I'll give you a clinical example of this. A second application would be whole genome sequencing, where the entire HIV genome is sequenced, and this typically uses NGS. Third would be long read NGS, where long strands of DNA are sequenced in one stretch. And finally, point mutation assays, where specific HIV mutations are detected using a lateral flow device. So first, to start with an application of SGS, shown from our group here. So in this case, an individual in the US acquired HIV on cabotegravir prep. Cabotegravir, as a reminder, is an INSTI inhibitor. But commercial Sanger sequencing did not reveal any mutations. So day 30 after they acquired infection, or infection was detected, single genome sequencing was used on their plasma sample to sequence 24 individual genomes, and this revealed 8% mutation frequency of the INSTI mutation Q148R and 4% mutation frequency of the INSTI A128T. And so this is just an example of where, because this was an individual who acquired HIV on PrEP, a technology to really see low-level frequency was of interest. Another example here is an application of whole genome sequencing. Whole genome sequencing is an increasingly important method, particularly with new data that off-target mutations could play a role in INSTI resistance, or with the introduction of inhibitors in newer drug classes like capsid inhibitors, which are not currently covered by routine clinical Sanger sequencing. So in this experiment, HIV was passaged in the presence of increasing concentrations of dolutegravir. This was a laboratory experiment done with cell culture, and mutations in envelope and nucleocapsid were found to reduce dolutegravir sensitivity. And so this is an example of a future application of whole genome sequencing that could be used to identify off-target genes in individuals failing INSTI-based ART. In long-read sequencing, long fragments of DNA are sequenced without breaking them up into smaller fragments, as was the case with NGS. Shown here is an example of long-read sequencing with nanopores. So nanopores are small holes in a synthetic membrane. The double-stranded DNA is enzymatically unzipped, then passed through the nanopore with an electrical current. Each base modifies the current differently, which can be read and translated into the order of bases. This has worked very well for SARS-CoV-2, but has some challenges for HIV due to homopolymeric regions that can cause some accuracy issues, although many groups are working on solutions to this. So finally, the OLA Simple assay is a method that can be used point of care. In OLA Simple, DNA can be obtained from a variety of sources, including DBS, or dried blood spots, PBMC, cell lysates, or cDNA that has been reverse transcribed from extracted RNA. The starting material is added to a dry PCR mix, resuspended in a buffer, and amplified. The unique aspect of this method is that mutation-specific complementary probes are ligated to the PCR mix, and the product can be detected on a lateral flow cassette to differentiate between wild type and mutant at each codon of interest. So here the bottom panel shows the analytical sensitivity of this method. Mixtures of 0, 15, and 25% mutant with 1,000 copies of HIV DNA were tested, and with each mutant shown, they found a significant difference in signal detected between wild type and 15% mutant. This assay is used for identification of specific drug resistance mutations. New iterations of the assay would be required, however, to add in additional mutations. So you can really only detect one mutation at a time here. So I've told you a lot about the current assays and emerging assays for drug resistance. So I'll conclude by summarizing the advantages and disadvantages of each method. So Sanger sequencing is the oldest method that's used. Some of the advantages are that it's lower cost for low sample volumes. It's very well established with established analysis and validation tools and a lot of software that's available. Analysis can be done manually or using publicly available databases. Some of the disadvantages, though, are low throughput, labor intensive, and a limited threshold for mixed base calling if that sensitivity is important. For short read NGS, there's high throughput for high sample volume. Due to this massively parallel reading, you can sequence multiple gene and regions and genes at once. Short read NGS is not that helpful for individual samples, and the analysis and validation tools are still under development, especially for low frequency drug resistance mutation detection in publicly available platforms. For long read NGS, it's high throughput simple workflow as well. The drug resistance mutation genome linkages are possible, which may be important in drug classes like INSTIs, where you want to see if there's accumulation of INSTI mutations, and there's de novo alignment versus reference guided. Here, too, the analysis and validation tools are still under development, and accuracy could be an issue for some specific regions in HIV. And then finally, point-of-care assays could be used, maybe particularly useful in resource-limited settings, and it is very advantageous for gene targets that have signature mutations like M184V or K65R for tenofovir. However, you do need multiple assays to detect mutations, such as for different pathways for INSTI, and you can't evaluate mutation combinations. So just a few conclusions and key takeaways here is that HIV drug resistance testing is an important tool for clinical management and population-based surveillance. Refinement of technologies are needed to expand HIV testing for INSTIs. Some useful advances could be amplicon Sanger assays. In the future, whole genome sequencing may be of greater interest for off-target genes and new inhibitors, and just making sequencing more accessible in various parts of the world will be an important advance. So I'd like to thank the organizers and acknowledge some of my colleagues at the University of Pittsburgh, including Carrie Penrose, Lou Halvis, and John Mellors. Thank you.
Thank you, Dr. Parikh. We do have time for questions. If there are any questions from the audience, please step up to the microphone and ask the question.
Thanks, that was very interesting. I have a practical question. What indications do you get to know that someone is in trouble on first line? Is it from a routine viral load? Do people feel bad and they come in and you do a viral load? And I guess the next question is, all my work is in low middle income countries, so I don't really know what happens here in the US or Europe. And what levels of viral load give an indication that maybe something is wrong? And did any of that make sense? Good.
Did I take it? Yep. So, excellent question. So, throughout the years, the answer to your question would have been different. There are kind of three thresholds. You could think about viral failure, which is measure viral load. You could think about immunologic failure, which is measure CD4 count, which is how we kind of measure the immune status of the person. And you can think about clinical failure. Someone comes in sick, as you were saying. So in the early days, when we had less lab ability to do viral loads or even CD4 counts enough, we would go by clinical. Then CD4 count came to play and we could do CD4 counts, and now, thank God, we could do viral loads. So these things, if you wait for someone to come in sick, they're going to be in more trouble than if you do viral load on a routine basis and find them in an earlier stage. So the answer to your question is we do viral testing routinely. Routinely differs in different places around the world. In the US, in my clinic, I usually do viral load testing for someone stable twice a year. If they're a little bit more trouble, I could maybe do it every 3 months, or if I want to, I can do it every month. In developing countries, guidelines are a little bit more strict, but meaning you could do it, let's say in the first year, you could do it more frequently, and then later on you can do it less frequently, depending on how the patient is doing. But in general, viral load testing availability has increased significantly throughout the world, which is wonderful, and we can identify someone earlier on. Does that make sense and answer your question?
Yeah, so what is the level, though? What is failure for viral?
Right. So that also differs in different places. I don't know if someone else wants to take that.
Maybe I can add to that, and it's often quite complicated. So the initial threshold was based on what viral load threshold would be practical in terms of doing drug resistance testing. So that initially determined it. And I think resource-rich guidelines usually use 200 because below that there's a bit of noise. I mean, that's based, I think, was a CID article, and Rami can comment on that. But there's a lot more noise below it. But any viral load that's unsuppressed is a concern, but you may not immediately act on it. So you would follow up and do another one. Now, even in South Africa, our guidelines have become more complicated recently because there's a bit of absence of knowledge of what to do with dolutegravir, because we don't expect resistance initially when they start on it. But still, in general, to do drug resistance, we need a threshold of 1,000 copies. So 1,000 is a definite hard action point. But any detectable viral load is now considered in our context. Anything above 50 is a concern. So these patients need to be monitored more closely. But we would not act and do a resistance test or transition them to another regimen before they hit this level of 1,000. So that's just a practical thing. But the problem is we do see resistance below that threshold. So we need to learn from our current experience and refine it over time. Thanks.
No, this is an excellent question. In some of the work that we do, we are trying to figure out how to transport blood from remote places to central labs. One of the issues is that in many countries, even that have resistance testing capacity, it's only really one main central lab in that country. And so DBS is still widely used because it's the most stable way to transport blood without dry ice. Once you spin blood into plasma, it really does need to be kept frozen in order to be able to detect HIV and do the resistance test. You lose sensitivity. So then you're trying to balance between, do I leave a sample at ambient temperatures and you lose that sensitivity because of degradation, or do I go with DBS, which maybe has a known threshold of sensitivity, which is going to be much higher than plasma? So it is an incredible challenge, and one that I think people are still trying to sort out. But with INSTIs, DBS is more of an issue because we do, as Dr. Van Zyl mentioned, need lower thresholds of viral load, and we know that the viral loads may not be as high, so it's a balance between being able to test that sample or not test it at all.
Can I add to that?
Yes?
I'd flip your question. Rather than saying stop DBS, go plasma, I would say develop a better filter mechanism to do it more accurately and in lower thresholds. And this is probably, a right thing to go. And I think people are doing it, and there are newer devices that are filter-based but hold larger volumes than DBS hold, for example, which is one of the limitations that we can go kind of much lower. So I would say let's develop something better rather than going to something that's more challenging.
Great, thanks. Any further questions from the audience? Yeah, if you could use the microphone, that would be great. Thank you.
Thank you so much for your talk. I learned a lot from you guys. And then my question is that nowadays people actually on the PrEP, on the risk populations, and then I found that all the news sounds very promising and very good, but some small fraction of people actually fail. And with your practice or research, those people on the PrEP of failure, the PrEP regimen, do they actually carry the mutation? Is that the one of the reason of failure, or simply just adherence is not that great?
Thank you for that question. That is also a big area of research that's ongoing, and I think the two reasons you cited are both important reasons. The most likely reason that someone would fail PrEP is adherence, that they didn't take it as prescribed or they had insufficient drug. Resistance is less of an issue as the reason for PrEP failure, but resistance can be an issue after acquiring HIV on PrEP, mostly through two pathways. One is if someone actually already had HIV and started PrEP because the diagnosis did not detect HIV before they started. That's the biggest risk of resistance with PrEP. Transmitted resistance is not as much of an issue, particularly now with the new INSTI inhibitors. The second reason would be if you did acquire HIV on PrEP and kept taking it without knowing you were infected, in time resistance could develop because the dosage of PrEP is insufficient to completely suppress HIV.
Thanks. Just maybe following up on that, maybe one of the other panelists would like to take this question, but what are some of the methods by which we can look at drug adherence in patients? Maybe Dr. van Zyl, you would like to address that?
I think that is a very important and exciting question. The problem is adherence measures in general are not objective, and there are electronic ways like memory caps and smart pills that people can use, but generally they're not available in resource-limited settings. So one needs more pragmatic approaches to know if patients are adherent. Something that is really very sensitive, but also very costly, is when patients take tenofovir, and one can do the same for other drugs with long interstellar half-lives like 3TC. One can collect dried blood spots, and then you can assess the concentration of tenofovir, especially in the red cells that are in, because the drugs can survive for long, a few weeks in those cells. So that can give you a longer time window of adherence. But because it's based on LC-MS-MS, so it's liquid chromatography and mass spectrometry, tandem mass spectrometry, these assays are very expensive to test the dried blood spots. Alternatives are things like, we have colleagues, Monica Gandhi and others developed lateral flow assays that one could do on urine that could also detect tenofovir. But the challenge with that is it has a very short window. It just tells you about adherence in the last few days. So, and also why would a patient now test this and not tell you that they haven't taken their tablets just before they came for their visit? So there's this desirability bias and what they call the white coat effect. So people might become more adherent just before they come. So it's really difficult to assess and monitor adherence. So, and there's lots of behavioral aspects to it. So it's a really difficult thing, but we don't currently have a good solution to something that's both sensitive, has a long window that it will tell you about adherence for a long period, and would also be affordable. So we're still looking for that. It's, if there's development in that field, it would be wonderful.
Great. Any other questions from the audience? Maybe one question for the panelists is, it's mentioned that drug resistance testing differs quite significantly in developed countries versus low to middle income countries. Would anybody like to comment on what the hurdles are for implementing more routine HIV drug resistance testing in LMICs? Maybe Dr. Parikh, you'd like to take that?
Sure. So we spoke of one of the challenges, which is really getting samples to their destinations in a form that preserves the sample integrity. I think some of the challenges is equipment availability, training, and really having laboratories that are that have the capacity to perform the testing and go through all of the steps needed. As you saw in some of the workflows, resistance testing can be complex workflows, so you need established laboratories that can have PCR clean rooms, PCR dirty rooms, the equipment. So once countries can establish these kind of laboratory infrastructure, that would really increase the possibility of resistance testing worldwide. Would you like to?
Yeah, and I think the other thing is skill set. Lots of these things are complicated, so you need to train people. You need to have people with those skill sets available in places, and the setup costs can be very high, as Dr. Parikh said. One needs, and there are smaller alternatives, smaller instruments, lower setup costs that are more attractive thing. But then also you have the bioinformatic needs, especially if you want to do NGS. Some approaches generate so much data, you need lots of computing power. And again, you need a bioinformaticist then to analyze the data. So one needs simpler solutions on that end, on the interpretation side also. So there's a lot of development. We're also involved. We're planning a project to try and solve some of those, to come up with some solutions for that. But it's a huge challenge.
Right, and just to add to, some of these newer technologies that may be more accessible are not yet cleared to be used clinically. So I think there's a long pathway to have a test actually used as a diagnostic for resistance, for example, or so that will take some time to bring the newer technologies out to the field, which is why I think Sanger sequencing continues to be the standard.
Maybe one last question for the panel. We're obviously hearing very good news about the long-acting injectables in the PrEP setting. And then more recently, there has been other good news about different HIV drugs in different classes with some good success stories there. But maybe Dr. Kantor, you would just like to summarize briefly what's on the short-term horizon for HIV, the new HIV drugs.
Yeah, sure. So I think, this is a race between the virus and us, basically, right? We develop a drug, the virus develops resistance. We develop a drug, the virus develops resistance. This is how it's always been. And I think that race continues, even though we now have this great drug class that we're learning. Well, actually, there might be more resistance than we think. So we have to continue to try to be ahead of this race. I would say this includes adding drugs to existing classes like we've been discussing, which is, we had first generation NNRTIs, we developed second generation. We had first generation INSTIs, there's now second generation INSTIs, et cetera. So we have to continue to develop drugs from existing classes. Then we have to develop new classes of drugs with new targets, and again, we mentioned some of those. And the third is that we have to develop new ways to get drugs to people. And long-acting is another revolution that is already coming into play. We have medications that we could give drugs to people by injections rather than daily. We can give it to them every 4 weeks or every 8 weeks, and then even right now every 6 months for different populations of patients. And we have the same concept in pre-exposure prophylaxis like we've been discussing with cabotegravir. So I think these are the trajectories that will continue, and the drug development must continue in on all these three categories in order to stay ahead of the virus the best that we can.
Thank you. Well, I'd like to thank all of the speakers today for really excellent presentations and a tremendous overview of different aspects of HIV drug resistance, and thank you all for attending.